foldable ultimately. Okay, so this is what it'll look like over the 4 macromolecules. So everything will be right here. You'll be able to study it really quickly. You'll be able to like quiz yourself. Okay, so I think it'll work out really well. So take your paper, whatever color you chose, and first thing, go ahead and fold it in half, hamburger. Like that. And you can crease it there. Okay? And you may want to clear your desk off. You won't need your computers anymore. You'll just need your scissors and your colored pencils. Okay, open it back up. Now you're going to take the top half and fold it down to that middle crease. And then you're going to do the same thing with the bottom. So take the top half and fold it down to the middle crease. And then take the bottom half and fold it up to the middle crease. So it should look like this. And then I'm going to fold mine in half the other way so I make a crease so I know where I want to cut. You guys with me? And then take your scissors and you're going to just cut on the top flap along that crease. And so you're making little flaps like this. Then do the same thing for the bottom. There you go. So it should be like this now. Everybody there? Okay, now get your pencil, and we're going to number these squares, okay? And that's how we're going to go through the slides presentation, so you'll know what to write where. And so, on your paper, the front of this flap is number 1, and just a little number 1 in the corner, okay? So just draw a little number 1 in the top left corner, or whichever corner. Just don't make it too big because we're going to write other stuff. Now, flip that up. This is going to be 2, 3. Okay, 2, 3. 2, 3. Two, 3. We got that? All right, the front of this flap is going to be 4. So the front of this flap is 4. Open it up, 5, 6. 5, 6. Stay with me. Bottom left, this is going to be 7. It's going to be 7. And then open it up. 8, 9. Okay? 8, 9. Last one, front of this flap, gonna be 10. 11, 12. 11, 12. All right, so we ready to fill it in now? So all of these, we're going to put, there's 4 major macromolecules, so this works out really nicely, okay? So each flap is going to be one of the 4 macromolecules that we talked about yesterday, and we're just going to kind of go through a little more details. The first thing I wanted to make sure everybody knows, all 4 of these are called polymers. polymers, and they're made up of smaller things called monomers, okay? So, for example, if we have a big thing of sugar, each little glucose is a monomer, and the big carbohydrate is a polymer. These are just like general words for these things. Poly meaning many, mono meaning one, okay? So, here we go. So, box 1, big. This is like a big label. You're going to write carbohydrates, which are the sugars, and we're going to draw a Dr. Pepper can, we're going to draw some bread, and we're going to draw some pasta. Okay? So on the front, in big letters, carbohydrates. And these are the sugars. And we're going to draw, you can draw your favorite pop. You don't have to draw Dr. Pepper, I guess. And then draw some bread, like a piece of bread, a slice of bread, or a loaf of bread, depending on your artistic abilities. And draw some spaghetti. Or your favorite pasta. I'm going to need to label my pasta because it doesn't really look like it's supposed to. And you may want to label if you're not going to remember that that's bread later. But hopefully you will. Are we ready for the second? This is probably the longest because I know there's drawings on this one. We good? And some of you who are way more artistic than me may come back and want to improve your drawings. So, in box 2, so open it up. This is what you're going to write in box 2, okay? The monomer for carbohydrates are called monosaccharides. What do we know that means? We've had this word already. What does mono mean?
Single sugar.
Single sugar, one sugar, okay? Monosaccharide is telling us one sugar. The major monosaccharide is glucose. Okay, that's the one we use. A disaccharide, what do you think a disaccharide is? 2 sugars. And the most well-known one is lactose. Where is lactose found? Milk, right? Dairy. Some of you cannot break down lactose. So you are lactose intolerant, right? Because you can't break the disaccharide. So it's 2 sugars together and you can't break that bond anymore. And so you can't drink milk, or it bothers you if you do. Polysaccharide, what does that mean? Many sugars, okay? And so we have a thing called glycogen. We store it in our liver. And plants have a thing called cellulose. And those are the big polysaccharides. And there's more, but these are just some examples. And really, maybe the most common and the most important examples. Another name we hear for plants is like starch, right? Also a polysaccharide. If you want to write it in parentheses, another word for monomer would be like a subunit, right? That's another way of saying it, is a subunit. Mine are way. not ready? Okay. Box 3. Function. And this is what we're going to put over. So in each of these, you're going to have like what they are in the front with pictures. You're going to have some examples, and then you're going to have the function. Okay? So really what you need to know in a nutshell. Function. Energy source. We talked about this yesterday. Carbs are our quick energy source, right? If you don't use it in 24 hours, you store it. But everything you can use in 24 hours, you will. And we burn these. We burn them in cellular respiration. Every cell in your body burns glucose. The other function is structural support. That cellulose is why trees can stand. It's really kind of their skeleton are these carbohydrates. That's how strong they can be. So energy for us, support for the plant. Okay, we ready for number 4? Okay, number 4, lipids, right? These are the fats. Lipids. These are our fats. So write lipids big on the front. Okay, and what we're going to draw on this one, we're going to draw some oil, right? We know that's an example. We're going to draw some butter. And I added an extra one that we didn't talk about yesterday. Honeycomb. Bees, like when they make the honeycomb, that's a lipid. Any kind of wax is a lipid, okay? So honeycomb is a good example of a lipid. So oil, I'm definitely going to have to label my oil. Butter. I'm so bad with like 3D drawings.
These are supposed to be 3D.
Well, I was trying to make my butter like, look like butter. It doesn't. It really, really doesn't. And then I'm drawing a little bee next to my honeycomb.
I just
It's too much water.
Where else do we see wax in nature? Natural wax.
candles.
Okay. Well, I'm thinking things that just like happen.
Earwax.
Earwax. That is a lipid. Okay. What do lipids do? What's the point of earwax? There's a point. There
It
is.
catches bugs.
Well, do you? Okay.
It's something with bugs. I just know that.
Well, it can't, but it catches foreign things that come into your ear. Okay? And so a lot of times a wax is like a couple things. Usually waterproof. Okay?
Is that why it looks
It's-
so gross?
Yeah. It's like a waterproof, and it also like seals off things and keeps things out. Have you guys, like when you have an apple, you know how you can scratch like the surface of an apple? Have you ever done that?
Yeah.
That's wax too. So a lot of fruit has a layer of wax around it to protect it from the environment.
Is that why you wash apples?
You wash apples because they've been on a trip for like 6 months.
You wash it.
You can eat the wax.
Oh.
I know a guy who like he runs the like the vegetable part of Walmart, and one time he told me apples can sit like a year. It can be a year since it's been off the tree.
Oh my gosh.
Yes, because they spray them with a chemical that keeps them from ripening, and then they spray them with a chemical to ripen them. So like apples can be off the tree close to a year.
Yummy.
Isn't that crazy? That's why we wash our
Chemical.
fruits.
Tasty.
Scary. Okay, the monomer. So again, we're talking about subunits, right? So the monomer for lipids is going to be fatty acids. So this is box 5. So your monomer is fatty acids. And one thing I want you to know about all lipids, they're all nonpolar. We talked about that, right? The oil won't mix with water because it's nonpolar. All the lipids, waxes, fats, oils, they're all nonpolar. That's what they have in common, okay? That's what they all have in common. And then what we talked about yesterday, there's saturated fatty acids that are solids, like butter. And there's unsaturated fatty acids.
Wait, so if you were to melt butter, it would,
But we're talking at room temperature. I mean, its chemical makeup is saturated. Now you can, I mean, if you get them hot enough, you can turn them all into a liquid, just like bacon grease too, if they're hot enough. But we're talking at room temperature, they should be a solid.
What if I heated it up?
how unsaturated?
It doesn't change its actual chemical makeup. And actually for this one, guys, I want us to draw something extra. So next to saturated, I want us to draw what it looks like. And so it has a carbon backbone. So this is your saturated one. And when it says it's saturated, it means it's saturated with hydrogen, which means every possible place has a hydrogen bonded to it. So that's what a saturated fatty acid looks like. It's completely saturated with hydrogen.
Does it matter how many C's we have?
No, I did 5, but an unsaturated, we're gonna draw that too. Unsaturated has a double bond. And when they double bond, they form little, like, kinks. It changes the direction. Now, you guys remind me, how many bonds can carbon make? Up here you can see it. How many bonds can carbon make? 4, right? So this carbon has already made how many? 1, 2, 3. You guys see that? And so it can only have 1 hydrogen around it. So this double bond counts as 2. Okay? And so we say this is completely saturated with hydrogen because you can't put any more hydrogen around it. This one is unsaturated, right? Because it doesn't have as much hydrogen around it. Now remember yesterday we talked about trans fats? If we like forced hydrogen onto this, it becomes margarine. Becomes, I can't believe it's not butter. Now, if it heats up, it loses the hydrogen and turns back into its natural form. Okay. All right, square 6. Some examples of lipids. Phospholipids. These are what make up the cell membrane. We're going to draw one of these in a second. Steroids. Okay, what do you think when you hear steroids? Strong, like taking steroids to try to, illegal steroids. Actually, vitamin D is a steroid.
Oh.
Okay, so not all steroids are bad. Now there are bad, like vitamin D. I'll show you. There are bad steroids, but they're not all bad. Okay, so steroids like vitamin D, estrogen, which is the female hormone, testosterone, which is the male hormone. Those are all steroids. Okay, and these are all lipids. And then wax, we talked about, is another example. Leave room because we're going to draw a phospholipid. Alright guys, next to your phospholipid, so this is what a phospholipid looks like. I just want you to have this in here. It's got what we call a hydrophilic head and two hydrophobic tails. What does hydrophilic mean? Let's start with the easier ones. What does hydrophobic mean? Anti what? What's hydro? Water. Hates water. Okay? So these are anti-water. So what do you think hydrophilic means?
Pro-water.
Pro-water. Likes water. Okay? So draw a little phospholipid. Okay, we ready for the next one? Hey guys, real quick, why don't you put your name on the back of your foldable just in case. If I were you, I'd tape it into my notebook, but I mean that's what we'll probably do, but just in case somebody misplaces it. Okay, we ready?
You look at Frankly.
Okay, here we go. Number 7, proteins. Number 7, proteins. So for this one, oh, you can't see my egg very good, but for this one, I thought we would draw a steak and an egg. Okay? So for proteins, do your best to draw a steak. A T-bone's always good, you know, you kind of at least have an idea of the shape. And then draw an egg. Maybe label these. I'm gonna get a little technical with this one too. this is number 8. I need to see something. Okay. Draw this kind of at the top because we're going to draw an amino acid under it. Okay? So leave yourself a little space. So our monomer here is an amino acid. And they join together. What?
Polypeptide.
To make polypeptide chains. What does poly mean? Many. And peptides are the type of bonds. Okay. We're going to draw an amino acid just to get you thinking about what their form is. So that when you get to AP Bio, you can recognize them really easily. Okay? So here we go. An amino acid, every single amino acid, there's like 21 in nature, okay? Every amino acid has the same basic structure. And so that's what I want you to know. So every amino acid starts with a nitrogen. And it has 2 hydrogens attached to it. This is called an amine group. Okay? That nitrogen is bonded to a carbon. This is called the alpha carbon. It's bonded to another carbon, which is double bonded to an oxygen and a hydroxide group. Every single one is like this. So, the only difference between them is they have what's called an R group attached right there. And there's 21 different R groups, which make 21 different amino acids. And so every time you see this NCC pattern, you know you're dealing with an amino acid and you know you're dealing with a protein. Okay? Always. That's what you're going to be dealing with. All right. Number 9. Number 9. Functions. Yesterday we talked about proteins do it all, right? They are the ones that are carrying out these important functions. And so, functions of your proteins. Transport. They move stuff in your body, like oxygen. They speed up reactions, things called enzymes. They give structural support. In fact, however much you weigh, if you weigh 200 pounds, 30 pounds of you is just protein. Okay? 15% of your body mass is protein. Considering you're mostly water, 15% is a big amount to be one thing. So 15% of your body mass is just little proteins. And they control cell growth. Really important. Okay, last group. You ready? All right. Last one, nucleic acid. Nucleic acid. And I had a horrible time finding pictures for this one, so the double strand, like this, that's DNA,
I'm just gonna double it, Candy, for now.
okay? And then I want you to draw a single strand that looks like this. That's RNA. Alright, DNA is double stranded, RNA is single stranded. So I do want you to draw both of those on the front of your nucleic acid. Huge. We got that? Number 11, the monomer. It's called a nucleotide. Nucleotide. And the nucleotide is made of three things: a phosphate, a sugar, a nitrogen base. And we're going to draw this too. Okay, so let's draw what. So this is a nucleotide. And the first thing we have is a phosphate group. And it's bonded to a 5-carbon sugar. Deoxyribose, or ribose is that sugar. And then it's bonded to a nitrogen base. Okay, a phosphate, a sugar, and a nitrogen base. That's a nucleotide. Okay, and last one. That's perfect timing. The function. 2 big functions for nucleic acids. Store and communicate genetic information, right? We talked about that yesterday a little bit. 99% of what makes us human, we're all the same, and it's all in our DNA. And then 1% is different between you and I, and that's what makes us look different, act different, right? The differences between us are that 1%. But all of it comes back to our DNA. When we look at our DNA versus a tree's DNA, we see that's where the difference lies between us. It's what is coded in our DNA. The second big function is a thing called ATP. Okay, it's a type of nucleic acid, and that's what gives you energy. It's what we make in our cells. So, oh, there we go. And we may write at least ATP. So store and communicate information. transfer. Everybody go back to your front page of your nucleic acid. Like with the DNA and RNA, add a little starburst, like energy burst, and put ATP in the middle. Add that picture to the front of your, okay? So you can remember that it does energy stuff too. So make a little starburst on the front. And put ATP in it. So hopefully you have something that looks like this, where you can see all your parts. Okay? And we're just going to use this to start learning these forms. We're going to do a lot more with it, right? But this is just a place to start. So I have tape up here. I think there's some tape in that drawer too. If I were you, I would try tape it into my notebook or staple it something so it doesn't get lost. Because even if it's in your notebook, as long as you tape it like this, you can still use it to study, right?